Well, hello everyone. My name is Santiago Mideros, and I'm a professor of plant pathology in the Department of Crop Sciences at the University of Illinois. In my laboratory, we study the effects of crop systems on the environment on plant pathogen populations. So how pathogen populations change based on the inputs, whether they are what we do or environmental inputs. And we use this information to develop improvement practices for management decisions or develop novel management decisions if that is at all possible. Today, I'm going to be talking about seedling diseases of field crops with an emphasis on Pythium, and I'm going to discuss a little bit about why Pythium. And the crops I'm going to be going to be referring to is corn and soybean. That is for two reasons. Number one, the multiple species of plant pathogens that cause seedling diseases or seedling blights on corn and soybean, and they can exchange, right? So the same strain is able to cause disease on both crops. And the corn and soybean agricultural system these days is understood as a single entity, right? Producers of corn and soybean rotation is what is used to the most extent in the Corn Belt. And in fact, according to the U.S. Census of Agriculture analyzed here by Suloaf and colleagues, the share of the corn and soybean agricultural system in the United States has increased, and showing here the data by area is close to 3/4 of all the acres planted on for grains or oil seeds are in the corn and soybean system, and that with an increasing trend, right? And this is by area, right? If we look at it by pounds of actual production, is actually closer to 90% for corn and soybeans on for grains and oil seeds. And I don't think I need to remind this audience about the importance of the corn and soybean system for industrial purposes, but it's also important to talk about how vital it is for food security. So, and worldwide, right? So the grain that's produced here not only stays in the States, but it moves around the world. So the challenge is to keep up with that productivity for industrial and food security while trying to generate perhaps better, more sustainable management practices, and as we move into the future. And one of the constraints for this corn and soybean system is these seedling diseases. And the seedling diseases, how they show up is first is poor emergence that we're seeing here. These are field trials where obviously there is a row of maize corn plants that are missing here. There's a missing plant here. And this poor emergence is going to lead into poor stands later on the season. Next, we will also see here is with soybeans, skips or yellowish or stunted seedlings that occur in patches or individually. Also, and this is visible when plants are a little bit older than these, and you can see it from the side, perhaps when there's like waviness on the rows that has been planted. That's an uneven plant height, another symptom. And other things you might see is wilting outright and leaf necrosis and scorching. This is the same, the roots of that same plant that here is shown clearly rotten. And as I was saying, this happens for both corn and soybean. If you look at the every single title of the last few slides, it is comes from straight from the compendium of corn diseases, and you can find the same descriptions for symptoms on the compendium of soybean diseases. It's just perhaps in different pages and different parts, but the descriptions are exactly the same. And one more typical symptom that I'm not showing here, but is important, is the damping off. And this is just looking at the seedlings more closely and early on when they are just starting to emerge and germinate. And there's that pre-emergence damping off when they seeds just do not even emerge. They die before they have come through the soil line. And then there's the post-emergence when they die shortly after. And this damping off occurs under high humidity conditions. This picture obviously is not high humidity conditions. I thought there was some humidity in that field earlier, but now it's all gone. But the damping off occurs under the high humidity conditions. And Well, the impact of seedling diseases showing here is like, again, field trial inoculated on this row with the red stake. This one was a control that's not inoculated, and well, obviously, very significant damage. Um, it's obvious there. So for corn, there are estimates that in 10 years the losses have amounted to 15 million bushels, and you can calculate how much that in money is, but it's a significant damage, but obviously changes by year for the money, for the monetary value. For soybean, there's even more surveys. A lot of more studies have been done on soybean. And in only 4 years, from 2006 to 2009, the estimates for losses were 40 million bushels. And the more reliable estimator is the ranking. I think about what are the most important diseases, and seedling diseases on soybean are usually ranked number 2 as the most important disease, just after soybean cyst nematode. Well, and that's yield reductions, right? So obviously a significant problem. There is also a significant effort that if the fields need to be replanted, and if the field is not replanted because it was too difficult to get out on the field again, or just the cost is too high for that, or the seed was over. There was no more way to get seed. There will be fallow areas in the fields, and obviously that causes significant issues. And obviously, seed diseases have been one of the main drivers for the increased use of seed treatments. And nowadays, in the Corn Belt, at least here in Illinois, most all of the soybean seeds and corn seeds that go into the field come coated with a seed treatment. So, and that's because that's one of the ways to manage this disease, right? And in fact, I think that the for corn production practices, the seed diseases were incorporated much earlier. The seedling the seed treatment was incorporated much earlier, and that's why probably some of the losses that I talked before were lower for corn than for soybean. But still, there is the significant issue of the cost of the seed treatment, and well, there has to be some environmental impact on putting so much fungicides on the soils. But so who causes these problems? And many pathogens. And there's over 2 dozen species of Pythium that cause seedling diseases on both corn and soybean. And again, the same species, the same strain can cause the seedling on both corn and soybean. We have tested that in the laboratory. For soybean, there's also Phytophthora, 2 Phytophthora species, especially Phytophthora soji, a very known pathogen, although it causes a seedling blight phase, but also a root rot phase a bit later. But it's part of the problem with soybean if we consider seedling diseases just happening early on the season. In addition to that, Fusarium is a pathogen of both corn and soybean, and Rhizoctonia as well. We have a few other pathogens on the corn side. Now, we know these are the causal agents, many of them, right? Several dozen species of Pythium, about half a dozen species of Fusarium are able to cause the problem. That's being studied from surveys about corn and soybean, more data again for soybean than for corn. And from those surveys, we also have a hint a bit which ones are the most important. And it's not completely known, because it's a little bit difficult to separate which one is more important than others, because depending on the media that gets used to try to recover the pathogens, you can target either the oomycetes or the fungi. They prefer different medias, and between Pythium and Phytophthora, they also prefer different medias. So it is not completely known which species will be the most important, but the hints are that Pythium and Fusarium are the most important, just considering the seedling blights. If you also consider the fact that Phytophthora soji causes a seedling issues, and later on root rots, Phytophthora are very important for soybean too. But then we have the Pythium and Fusarium as the most important oomycetes and fungi causing those seedling blights. And So this also comes from a survey we did in Illinois, and we haven't published this data yet, though it was some time ago. We did. The reason was we were looking for Phytophthora, but we got a lot of Pythium. And what I'm showing here is like areas or the location of the samples that we got, either soybean plants that were affected by a seedling blight or soil samples. And you notice that there's a lot of green there, and then that was a lot of Pythium ultimum species that we identified using microscopy and DNA. Yeah, there's a lot of Pythium. Even when we were looking for Phytophthora specifically, we collected a tremendous amount of Pythium. That's why made us consider looking more into this issue of Pythium as a major component of the seedling diseases for both corn and soybean. So obviously there is not a lot of depth on these samples, and the more you sample, the more species you get. So this is not a comprehensive survey by any means. So what do we do? What do people do about managing seedling diseases? Obviously, we start with the basics, which will be high quality seed. After that, as I said, seed treatments. We apply a tremendous amount of seed treatments now, and they are very effective. We'll talk about that also for both corn and soybean. For soybean, there is the added resource of resistance to Phytophthora that can be also used. There's at least two forms of resistance that are known, and several resistance genes. I'll talk more about that a little bit. For both corn and soybean, obviously improving drainage has many benefits. Among them is the management of seedling diseases. I mentioned already, replanting might have to be done, and people do replant in many cases. And there's also more of an aspirational avoid cold temperatures, but the reality is that producers, farmers do plant when they can, and they do try to plant earlier these days because there's a lot to be gained with on the yield front. So we need to adapt to that, and perhaps that's why we're trying to apply more seed treatments if we're planting earlier. So I mentioned resistance was a way to manage at least Phytophthora on soybean, and which is one of the most important issues for soybean grown in the Midwest. And I had a grad student that, on that survey before collect all those isolates of Phytophthora we have gotten, and looking at those from Illinois, and look at them, test them again, a panel of soybean lines that have different resistance genes. And what I'm showing here is how many of those isolates, 100% in this case, were able to cause disease on a plant. This is the control, the RPS, that doesn't have any resistance gene. So most of those Phytophthora isolates are able to cause disease on that soybean plant, right? On the seedling phase. This was seedling assays on a greenhouse, and well, you notice that some strains such as that some genes such as RPS 3B, 4, 6 are very effective, right? 3B here allows less than 25% of the isolates were able to cause disease, and 4 and 6 even better, right? So you might want to avoid resistance genes 1B, 5, and 7. Even 1K, that was a very useful gene in a few years, nowadays gives less than 50% chance of success against Phytophthora, right? So that is with resistance for Phytophthora zoydes. There is also seed treatments, as I mentioned. There's many seed treatments, many active ingredients these days that can be used on that. What we need to keep in mind is that different active ingredients are effective against some pathogens more than to others, right? So for example, those metalaxyl, mefenoxam are targeting Pythium and Phytophthora, the oomycetes or the water molds, while ipoconazole, the azole fungicides, and many others are more of a target to Fusarium. So if we're trying to manage the seedling diseases, we have to have a seed treatment that incorporates a couple of these active ingredients, right? One from each group. There's others, right, such as the strobilurins with azoxystrobin that tend to be good against both fungi, the true fungi such as Fusarium, Rhizoctonia, but also have some effect against the oomycetes, Pythium and Phytophthora. Which is great. The problem is that these strobilurins tend to be, we need to be careful with the strobilurins because the pathogen can develop resistance to them. So there again, one way to manage that would be to put it in combination with another active ingredient. So again, another reason to have strobilurin plus something else, right? Nowadays, these seed treatments do not come only with fungicides. They also have insecticides, nematicides, and these days biologicals. That's something also that we're trying to study and get into a little bit more. So important to have at least a couple active ingredients from each group so that there is a comprehensive management of seedling diseases. Now, we did some tests actually with those Pythium isolates that collected in Illinois a few years back, and Danielle, my grad student, also tested several active ingredients for their effectiveness against those Pythium species, and there's some interesting discoveries on that is like, for example, what I'm showing here is how much of the doses of a active ingredient is needed to be effective against a specific isolate of each one of these species. And this is the number of isolates. So for example, here we have Pythium ultimum. We have a couple isolates that we need very low doses of azoxystrobin, for example, to be effective at stopping them. The same for other, like a couple of 3 strains of Pythium disoticum, for example, there. But then there were two strains of Pythium vexans for which this azoxystrobin was just at the doses we tested was not effective, right? Different reactions to a different active ingredient. Here, ethaboxam, for example, was very effective at stopping that Pythium vexans, but not very good against Pythium aphanidermatum here on the red, with very high doses of that fungicide. Not that good, right? So the efficacy of fungicide varies by pathogen species. Similarly to what I was just showing for resistance to Phytophthora zoyi, right? So there is not, these are not silver bullets, right? So they don't kill all the pathogens that are there, but they are very effective against one thing or another. Now, so we have a lot of management techniques here. So we wanted to conduct some independent tests. This is something we did some time ago with funding from the North Central Soybean Research Program on field trials, and we had replicates in multiple locations in Illinois, and we combined with some data that Alison Robertson had and her colleagues had also obtained from Iowa. And well, yeah, the seed treatment was very effective, right? In this case, it was the active ingredients included ethaboxam, metalaxyl, and ipoconazole as far as the fungicides and others. Very good at reducing disease. Also, in fact, there was better yield, right? The yield was protected, and on the field sites that had high disease pressure, we could see better yield on the seed-treated plots. There is also the RPS genes were also very effective, but on some sites and some of the locations. And again, this goes back to which strains would have been there present, and if the RPS gene was well paired to the Phytophthora zoyi strains that were present there. And as I mentioned, for Phytophthora associated, there is also a quantitative resistance that can be used, and was also very effective on those trials. So we want to see some more details on how these trials worked on managing Phytophthora root rot on soybean. There's more details on that paper, but yeah, seed treatments work, resistance works. It's basically the bottom line in there. I wanted to go back a little bit to the management and highlight the fact that for corn, we don't have that extra tool of resistance here, right? We're missing that. There is resistance to Phytophthora, at least I said there is the RPS genes, and there is like the quantitative resistance, also called tolerance against Phytophthora. And there is at least academic studies on resistance to Pythium, right? So there are some papers out there. I don't know that has been deployed into commercial agriculture here. This resistance to Phytophthora, it is definitely deployed and is accessible to any grower. But there's studies, at least people are starting to work on Pythium resistance on soybean, but not so much on corn. So we got some money from the National Science Foundation to start working on that area, especially because we were targeting trying to control multiple diseases at the same time. And that's what we're trying to do. So we have a graduate student that started looking into that. And what he did was first an in vitro screening. So he put a diverse panel of maize lines, so very diverse maize germplasm, and he put them to grow on a Petri plate that also contained Pythium, right? This was grown in an incubator for a few days, and then he measured the radicals, and he standardized the radicals to the non-inoculated controls, because although each maize line has a slightly different growth, so some lines have naturally larger or bigger roots when they are just germinating than others. So we had to control to standardize everything to the non-inoculated controls. And what he found was promising, that for some lines, such as the CML277, was very susceptible. You see, when inoculated with Pythium, in many cases, it did not grow at all, well, compared to how it would look on the Petri plates that didn't have Pythium. But others, like this H100, were actually able to grow even on the Petri plate that had Pythium. So the way this looks on the full set of lines was he identified some lines here on the green that were more resistant, and these are all the maize lines, and this is percent of radical lengths, right? So again, it's standardized to their control. So 100% would be where the radical of those maize lines were the same size as those grown on the Petri plates that didn't have Pythium, right? Side by side comparisons. And yeah, so some lines were a lot more, well, I'll say more moderately resistant here on green. Some were moderately susceptible, and some were much more susceptible to Pythium. But of course, this is in vitro plate. Can we make it a little bit more realistic? And so then what Harrison did was develop this assay for greenhouse experiments, where he will grow these corn plants, different maize lines on containers, and then he will flood them so that to mimic high humidity conditions that are conducive to specifically to Pythium and some on corn. And he collected, in this case, also the radical lengths and radical weights, but also collected a percent of disease severity, showing here like high disease severity was an 80%, for example, and very little disease severity being at 0%, right? So, and again, this was, we had to do one tote that has the inoculated containers with corn, with the corn and the and Pythium, and some that didn't have any Pythium, so that we can compare side by side each trait to the non-inoculated control, right? So the treatments really got a lot more disease severity, shown here, versus the standards that didn't get inoculation, that had lower disease severity, right? So under those conditions, if you remember that line I mentioned before, CML 277 was still very susceptible, as shown here on the disease severity. So in this case, 100% is those seedlings are not even germinating on those containers that had Pythium on the soil. Right, so it's 100% disease severity compared to other lines that were way more resistant, such as CML 103 or that H100 that was over here, a little bit more variability in this case. The colors are based on the in vitro ranking that I showed before, whether it was morally resistant or more morally susceptible or susceptible, right? Again, CML 277, looking at the root masses standardized, 100% being equal to the control. CML 277, very little root mass compared to the CML 277 when it was not grown with Pythium. Very susceptible, much more than most of the other lines. So yeah, let me just go slowly here. That is on a greenhouse trial. So he grew it at least 3 times, and getting some consistent results on some lines are more resistant, some more susceptible on corn to Pythium. Now, I mentioned there is several dozen species of Pythium that cause disease, and so any form of resistance that we have needs to be useful against several strains, right? So that we were talking about that, and then we thought, well, what we need to do is now test how resistant these resistant lines we're identifying, or susceptible lines are to other species, right? And that's what he did. And then again, collecting data on the percent of root necrosis, on the percent of on the length standardized to their controls, and the masses standardized to their controls. And he's repeated these experiments a couple times, again with consistent results. We're getting several maize lines. In this case, we're 8 maize lines, and there's consistently differences for the multiple traits that are being evaluated for the maize line. So we're seeing different levels of resistance. There is unsusceptibility in maize lines. The next thing is this Pythium species came a little bit significant, at least for some of the traits. And here showing us the length, the percent length for the standards and all the data accumulated. And what this means is that some species, so in this case, Pythium aphanider was a little bit more aggressive than other species, such as Pythium ultimum here on purple, right? They are able to reduce more the length of the roots than other species, such as Pythium ultimum here. This again is under the greenhouse conditions that we were growing them. But the exciting thing is that maize line by Pythium species interaction is not significant. So what this is telling me is that those levels of resistance to the 3 different Pythium species, even if they are one is more aggressive than the other, so but the ranks of those maize lines are similar, right? So they are not just scrambled. So suggesting me that the resistance works across multiple species, at least the resistance that we're looking here, or at least against these 3 species, right? So what's next? Well, Harrison is gonna keep working on putting this into a research paper, but what we want to do is identify those genes that are conferring resistance to multiple species. And the issue of environmental conditions is also going to come into this, and that's why we want to look at it from a complete disease triangle perspective, right? So it is known that different species of Pythium are more aggressive under different environmental conditions, specifically temperature, right? So we want to start trying different temperatures and see how they affect the pathogens. We would also like in the future to start looking into more of the whole microbiota, or that microbial seed bank that is on the seed bank itself, on the seed bed, right? So there is multiple pathogens, multiple beneficials, multiple neutrals. That's why I'm trying to look at it from more of a systems perspective, and how the environment affects that microbiota, which is pathogens, beneficials, and neutrals. The environment also changes, right? We're doing this rotation of corn and soybean. I want to see how that is changing this microbiota, hopefully, for the future, so that we can come up with better ways to manage these seedling diseases, or like ways to reduce this component of the pathogens on that on those soils. And already talk about the host. The idea is to identify what are those resistance levels that are start developing it also for corn, and try to provide avenues for a new tool for disease managing these diseases of seedlings, especially on corn. See if we can also work on a system level with the soybean rotation. All right, so that is what I have. The work, especially on the Pythium resistance that I talked about, is from Harrison, shown here on inoculating some fields, and Maracron has also been instrumental in that work, also continuing with that Pythium characterizations. The original strains were collected by Juan Pablo Granda, Daniel Cerritos, and Xunyang Juan, who did some of the initial characterizations, and we're continuing with that from the Pythium perspective. As I say, the project back there from the North Central Soybean Research Program was aimed more at Phytophthora. We encountered all these and they worked on that, and they published those results, but we also found all those Pythiums, and we also wanted to start looking at the Pythium strains now, which is what we're doing as we go forward with this Pythium resistance funded by NSF. So that is what I have for today. If you have any comments or questions, I will appreciate if you shoot me an email. And yeah, hopefully we learned something about how to manage seedling diseases on field crops, and something about what's coming down for the future tools for management. Thank you.